All right, so here we have bill analysis here in Texas, right? Health and Human Services, Senate Research Center, texas.gov, author, sponsor, statement of intent. There is growing concern that meat products for human consumption may come from livestock that have been injected with mRNA technology. This technology is new and long-term effects have not been studied. This bill would require that all meat food products that are sold in the state that are derived from livestock that have been injected with mRNA vaccine material include a clear and conspicuous label indicating the product contains mRNA vaccine material. The bill would define mRNA vaccine material as a substance included in a vaccine to stimulate the production of antibodies and provide immunity against disease by reducing messenger ribonucleic acid mRNA that corresponds to a wild protein. This bill is not intended to prohibit or discourage the use of mRNA vaccine usage, but rather to give consumers the ability to make informed purchasing and decisions until long-term research has been conducted on the transmission of mRNA technology to animal products and the benefits or risks of human consumption. Informed consumer choice must be maintained. As proposed, SB 2632 amends current law relating to required labeling of meat food products that contain mRNA vaccine material. Rulemaking authority. Rulemaking authority is expressly granted to the Executive Commissioner of the Health and Human Services Commission, Section 3 of the bill. So this bill. Section by section analysis. Section 1 amends Subchapter D, Chapter 431, Health and Safety Code by adding Section 431.085 as follows. Section 431.085 required notice on meat food products containing mRNA vaccine material. Defines livestock, meat food product, and mRNA vaccine material. Defines, prohibits a person from selling in this state a meat food product derived from livestock injected with an mRNA vaccine, unless the product's label includes a clear and conspicuous notice that contains mRNA vaccine material. Section 2 amends Subchapter C, Chapter 433, Health and Safety Code by adding Section 433.0415 as follows. Labeling of meat food products containing mRNA vaccine material defines mRNA vaccine material. Prohibits a person from selling in the state of meat food product derived from livestock injected with an mRNA vaccine, unless the product label includes a clear and conspicuous notice that the product contains mRNA vaccine material. Section 3 requires the Executive Commissioner of the Health and Human Services Commission, as soon as practicable after the effective date of this act, to adopt rules necessary to implement the changes in law made by this act. Section 4, effective date November 1st, 2023. So this was less than a year ago. I mean, for all intensive purposes, this bill is stating that they will not allow the sale of mRNA treated meat on the market here in Texas. .gov, right? Capital. So, whether this is passed, whether this is being considered, I don't know. You know, you take a look at these bills. You know, I mean, got us stated clearly somewhere on the progression of this judicial endeavor right here, which does doesn't really seem to state much more than what seems to be a proposal of some sort. Let's take a look at it again. Health and Human Services. Okay. Research Center. Author responder statement. Bill would require that all meat, food products sold in the states that are derived from livestock that have been injected with mRNA vaccine to include clear, conspicuous label indicating the product status. Yeah, that's what it keeps on reiterating. As proposed, amends current law relating to required labeling of meat, food products that contain mRNA vaccine material. As proposed, SB 2632 amends current law, right, relating to required labeling of meat products that contain that law relating. Well, then we would have to find out what the Health and Human Services Commission has to say about Section 3 specifically as of currently, or as of recently, because this really is going to be a matter of yeah. So, let's go ahead and take a look at this thing, see if we can find it right. Here we go. All right. Let's see. Okay, so let's take a look here. Um, by Hall et al. The Senate filed May 10, 2023. May 11, 2023 reads first time and referred to Committee on Health and Human Services. Favorably committee stated by the following yes vote. Nays may substitute. Bill to be entitled an act related to requiring labeling of food containing an mRNA vaccine material, be it enacted by the legislature of the state of Texas, Section 1, Subchapter D, Chapter 431. Health and Safety Code is amended by adding Section 431.085 to read as follows. Section 431.085, required notice on food containing mRNA vaccine material. Required notice on food containing mRNA vaccine material. In this section, mRNA vaccine material means a substance included in a vaccine to stimulate the production of antibodies and provide immunity against a disease by introducing messengers of ribonucleic acid mRNA that corresponds to a viral protein. It says mRNA vaccine material, right? And it defines it too. So, a person may not sell in this state food injected with an mRNA vaccine as defined, right? An mRNA vaccine as defined. Um, unless the food's label includes a clear and conspicuous notice food contains mRNA vaccine material. Now, it should probably say mRNA vaccine technology, really. Not just materials, but mRNA vaccine technology. It's like, by all means. So that's what needs to be actually put on the label. So Section 2, Subchapter C, Subchapter 433, Health and Safety Code is amended by adding Section 433.0415 to read as follows. Labeling of meat, food, products containing mRNA vaccine material. In this section, mRNA vaccine material means a substance included in a vaccine to stimulate the production of antibodies. In this section, mRNA vaccine material means a substance included in a vaccine to stimulate the production of antibodies. Let's see. Provide immunity against disease by introducing messenger ribonucleic acid mRNA that corresponds to viral protein. But I should finish reading this. Person may not sell this state of meat, food product derived from livestock injected with an mRNA vaccine unless the product's label includes a clear and conspicuous notice that the product contains mRNA vaccine material. As soon as practicable after the effective date of this act, the Executive Commissioner of the Health and Human Services Commission shall adopt rules necessary to implement the changes in the law made by this act. Section 4 of this act takes effect January 1st, 2024. So this was not right? This was not in practice, or I guess, I mean, I'm suggesting this is in practice now, but this wasn't in practice before this year, so whether or not we ate meat that had mRNA vaccine material, as stated, right? Which, by the way, I guess we should be looking for that that label and what it looks like. And it should say mRNA vaccine technology, to be clear. That's what it should be saying. All right. All right, so here we go. So, U.S. If you Google clone meat in the food supply list, right, you'll see that the U.S. Food and Drug Administration has approved the use of meat and milk from cloned cattle, pigs, and goats. Approved the use of meat of milk from cloned cattle, pigs, and goats, and from the offspring, right? The offspring. More importantly, the offspring of the clones of any species traditionally used as food. It said that such meat and milk was as safe to eat as good as food from conventionally bred animals. FDA approves use of cloned animals from food and considered to be as safe to eat as food from conventionally bred animals. I mean, I guess the FDA approves cloned meat to be grass safety, right? Um, generally uh, preferred as safe, or yeah, so on and so forth. So I guess that's for just our general information right there. FDA approved uh, meat and milk. So your cheese, so on and so forth, your yogurt, from cattle, pigs, and goats. But the offspring, right? So pretty much the progeny of this these clones uh, that are being implemented into just being, you know, dispersed into the the, the livestock, just being, genetically mixed into our food supply, because the offspring of these clones are going any species traditionally used as food, right? So that such meat and milk was as safe to eat as food for conventional bred animals. So before I go any further, this should be on the labels of any food. That goes without saying, right? Obviously, if we're eating meat from cloned animals, then that should be on the label. Besides the mRNA technology or materials, which, by the way, we would like to know what those are. Um, the U.S. Food and Drug Administration has approved the use of meat and milk from cloned cattle, right? Pigs and goats, and from the offspring clones of any species traditionally used as food. It said that such meat and milk was as safe to eat as food from conventionally bred animals. The FDA said that its conclusions agreed with those of a report from the National Academy of Sciences in 2002 and had been peer-reviewed by independent scientific experts in cloning and animal health. Scientific experts in cloning and animal health. Okay. Independent scientific experts, right? So, anyways, there was insufficient information to reach a conclusion on the safety of food from clones of other animal species, such as sheep. The FDA said it recommended that the food from clones of animals other than cattle, pigs, and goats was not introduced into the food supply. And it recommended that food from clones of animals other than cattle, pigs, and goats was not introduced into the food supply. So there was insufficient information to reach the conclusion of the safety of food from clones of other animal species such as sheep, the FDA said, and it recommended that food from clones of animals other than cattle, pigs, and goats was not introduced into the food supply. So pretty much they're saying that, everything else besides cattle, pigs, and goats probably not a safe or a good idea, but um, you know, by all means, you know, let your natural sensibilities, take a hold and let you know that um, There's an elephant in the room, but not an elephant. It's clones. So the U.S. Department of Agriculture will convene stakeholders to discuss efforts to provide a smooth and orderly market transition as industry determines next steps with respect to the existing voluntary moratorium, the FDA said. The Agriculture Department said that it has encouraged technology providers to maintain their voluntary moratorium on sending milk and meat from animal clones into the food supply during this transition time. Despite the FDA approval in principle of meat from cloned cattle, despite the FDA approval in principle of meat from cloned cattle, pigs, and goats, in practice, clones are not expected to enter the food supply. Practice clones are not expected to enter the food supply, the FDA said. They are rare and expensive, and the U.S. Agricultural Department estimates that most of about 600 cloned animals in the United States are used for breeding. Their offspring, however, produced through ordinary sexual reproduction, are likely to be used for meat and milk. So there you have it. The two largest U.S. cloning companies said they would keep a register of all their cloned animals so that food companies can identify them when they enter the food chain. According to the Environment News Service, 2007, cloned animals to be tracked for food processors. The agricultural department said that cloning was another breeding technique, compared it with other assisted reproductive technologies such as artificial insemination, embryo transfer, and in vitro fertilization used to produce superior animals for milk, meat, or breeding purposes. Fertilization used to produce superior animals for milk, meat, and breeding purposes. So the FDA FDA said, right, that you know uh, the whole purpose of them making these clones was so that way they would be likely be used for meat and milk. So cloning was another breeding technique. Paired it with other assisted reproductive technologies such as artificial insemination, embryo transfer, and in vitro fertilizer used to produce superior animals for milk, meat, and breeding purposes. Uh, Once again, right? I mean, trying to facilitate something that is not part of the criteria. mRNA technology is not vaccines, right? And cloning is not another breeding technique. So the FDA released documents outlining the agency's approach, risk assessment, risk management plan, the guidance for industry, Drafts for these documents were released in December 2006, and since then the risk assessment has been updated to include new scientific information that reinforces food safety conclusions, the agency said. No special labeling will be required on meat or milk from cloned animals or their offspring, because food derived from these sources is no different from food derived from conventionally bred animals. Should a producer express a desire for voluntary labeling, for example, this product is clone-free, it will be considered on a case-by-case basis to ensure compliance with statutory requirements. The labeling be truthful and not misleading, DA said. Arthur Kaplan, director of Center of Bioethics at the University of Pennsylvania, said in a blog that food is about emotion. The food industry is not going to like the emotions surrounding cloning. He said that U.S. consumers were wary of cloning, in part because films presenting clones as monsters, which had made them forget that cloning is nothing more than artificially creating twins. Every drop of wine we drink comes from cloned grapes. The Union of Concerned Scientists, the Consumers Union, the Consumer Federation of America, the Democratic Party Senator Barbara. Yeah, okay. So, which was made after it was received more than 3 comments from the public rights group, condemned the FDA's decision. Okay, so the Union of Concerned Scientists, Consumers Union, the Consumers Federation of America, the Democratic Party Senator Barbara Mikulski of Maryland, and animal rights groups condemned the FDA's decision, which was made after it had received more than 30,000 comments from the public. Wayne Pacelle, president of the Humane Society of the United States, said that the FDA had failed to take into account the animal welfare implications of cloning. High rates of mortality and birth defects and diseases in cloned animals. He also said that the meat and dairy industries haven't even been clamoring for cloning opportunities. It's been venture biotech companies and their allies in the for-profit branch of academia that have been doing harm to animals, developing clones as the latest new money-making scheme. Basically, yeah. Um, biotech, biotechnology. So, um, yeah, there you have it. I mean, what's my comments on this, really? I mean, FDA approves use of all cloned animals for food, but they went ahead and stated that they actually, you know, uh, have it in they they practice it. They practice it. They have it in practice. They have full legislature and production, ethics and values for this, and has been in use, has been in the food supply in use as far as um, food and well, not Food and Drug Administration, Food Administration ethics is concerned. So, um, if you want to ask yourself, have you eaten cloned meat? Well, the likeliness is that more than likely, well, the likeliness is yes. Um, is there a causal correlation between this and cancer? Well, that would require further research, but I mean, Arthur Kaplan, director of the Center of Bioethics at the University of Pennsylvania, said in a blog that food is about emotion. I mean, like, this is a director of the Center of Bioethics of the University of Pennsylvania, right? And and this is what was stated. Okay, well, that blog isn't there, but this scientific literature from 2008 is January 26th. So I guess what are my final thoughts on this? Um, I don't have any final thoughts on this at all. It's just, I'm gonna contact the Union of Concerned Scientists, the Consumer Union, the Consumer Federation of America, the Democratic Party, and see who cares about this, right? Um, Animal right groups. Uh, I, I think that you want to contact somebody else besides just animal rights groups. I want to contact um, generally every major food distributor and present both to Congress and every metropolitan area across the world, I would say, right? To your city council, public square, your city hall, um, governor, and discuss with them to put cloning labels on clone meat, since they're meat that's been cloned from animals that, generally speaking, the FDA um, has not really decimated to us, like the word decimated, disseminate. Very good. It hasn't disseminated to us the science or information around cattle, pigs, or goats that have been cloned, and Yeah, I mean, I'm kind of lost at words because I mean, what we're talking about here is that they're saying it's like, well, they didn't put meat, cloned animals into the meat supply, but they did put their offspring and everything else into the meat supply, into the food supply. So it's like, for all intents and purposes, you did put clones into the food supply, didn't you? It's like, well, FDA approves use of cloned animals for food. Um, should say something more like the FDA approves use of cloned animals for food and has uh, put into production and practice, which, I mean, now I'd have to look for the documentation as far as the ethics of FDA's ethics of the FDA's ethics of practices of cloned meat and milk. It's CPI. All right. Okay. All right. So, cloned animals products in the human food chain, right? FDA should protect American consumers. Yeah. Um, abstract. Animal cloning is complex process that lets one exactly copy the genetic or inherited traits of an animal. In 1997, Dolly the sheep was the first animal cloned, and since then, scientists have used animal cloning to breed dairy cows, beef, cattle, poultry, hogs, and other species of livestock. Cloned animals are highly attractive to livestock breeders because cloning essentially produces an identical copy of an animal with superior traits. The main purpose of cloning livestock is more focused on efficiency and economic benefits of the producer rather than the overall effect of cloning on an animal's physical and mental welfare. The focus of this article is threefold. First, the science behind animal cloning is explained, and some potential uses and risks of this technology are explored. Second, FDA's historical evolution, current regulatory authority, and limitations of that authority is described. Lastly, a new regulatory vision recognizes the realities of the 21st century global markets and the dynamic of evolution of scientific discovery and technology. The name of this one is Cloned Animal Products in the Human Food Chain. FDA should protect American consumers. Uh, yeah. I'm not seeing the actual document. Animal cloning, consumption of its byproducts. I mean, look at that, but I want to find this. Let me see. Okay. All right. Animal cloning is a complex process that lets. So let me see if this thing's complete, because otherwise, uh, it's one of those. All right. Well, I have to register on some of these websites to go ahead and get access to some of these medical journals, but let me see if I can find anything that's similar to that one right there, because that one seems to be right. Okay. Uh, Reviews of meat and milk and clones. Right. But okay. Study on current risk assessment and guidelines on the use of food animal products derived from cloned animals. All right. Um, Highlights. I mean, there is no evidence that meat and milk derived from cloned animals or progeny pose a risk to food safety. For four principles of safety assessment for meat and milk food derived from cloned animals suggest to establish the guidelines. Four principles of safety assessment for meat and milk derived from cloned animals suggest to establish the guide for the guidelines. Four principles are similarities. Yeah, okay. All right. Four four principles or similarities of chemical composition, reproductive effects, toxicity, and allergic reaction. If there are no differences in the four safety assessments, meat and milk derived from cloned animals are safe. I guess in comparison to natural meat, right? Given the meat is natural itself, I mean that's not just another GMO, which I guess would I don't know, man. I don't know. I mean, we're dealing with mRNA and clone meat, so at what point does this thing become GMO? It's like someone will probably come up with an answer. It's like, oh, just started doing it in the 70s. It's like, all right, well, anyways. The author widely surveyed and analyzed the food safety issues, ethical issues, permits, and approval of animal products from animal clone by somatic cell nuclear transfer worldwide. As a result of a 2-year survey, the author found that there is no evidence that meat and milk derived from cloned animals or the progeny pose a risk to food safety in terms of genotoxicity or adverse effects or allergic reactions. Most countries have not approved meat and milk derived from cloned animals. Most countries have not approved meat and milk derived from cloned animals. establish the guidelines, the authors suggest. The author, right, suggests 4 principles of safety assessment for meat and milk derived from cloned animals. The 4 main principles for safety assessment are similarities of chemical composition, adverse reproductive effects. They're reiterating the same thing again, so on and so forth. Go ahead and get to it. Biologic process of producing genetically similar animals. Uh, it's identical, but it's not saying similar. Okay, so it's the most common animal cloning technology. Like, I mean, this is the kind of stuff you actually put under scrutiny here. This is ridiculous, and this is the kind of thing you gotta go ahead and actually, put your magnifying glass and, I guess, you know, Sherlock Holmes pipe really works for you, then, by all means, put on a Sherlock Holmes outfit or whatever. But anyways, because somatic cells can be easily obtained, cultured in vitro, cloned animals are highly attractive to livestock breeders because cloning essentially produces an identical copy of an animal with superior traits. Nonetheless, safety of meat and milk derived from cloned animals and ethical issues in animal cloning are still controversial topics. In 2002, the National Academy of Sciences and National Research Council report commissioned by the FDA concluded that there is no evidence food products derived from adult somatic cells clones or their progeny pose a food or safety risk. The U.S. government has released a draft proposal declaring they're just reiterating the same thing. There's no scientific documentation here, which it's actually like established. Say materials admitted. Okay, surveyed more than 100 studies published in the past 100 years relating to the link between the animal products derived from cloned animals. The study included electronic searches of the PubMed. Yeah, okay. Risk assessment studies on animals from cloned animals. Extensively surveyed the risk assessment studied. Okay, the guidelines. Guidelines. Definition of meat and milk derived from animals cloned by SCNP for the guidelines. Cloned animal is a creature representing a genetically identical copy of an animal because of SCNP. And the definition of cloned animals include the progeny, which is definition of cloned animals the progeny, which is the children, grandchildren of the cloned animal. Uh, the definition of animal products derived from cloned animal are meat and milk derived from progeny. Second, third generation of the cloned animal, not the cloned animal's lifetime. First generation, definition of the progenies. Let me go ahead and see what else is going on here. Before and after slaughtering, the first step is analysis and measurement of the animals in the farm before slaughtering. Okay, come on. Establishment of the tracking system from the farm to the market, as mentioned above, determining where animal products come from. Clone difficult because clone animals produced by SC, okay, I guess SCNT or from non-clone animals is difficult because clones and cell donor animals share exactly the same genetic information. Cloning does not involve any genetic modification. Therefore, enforcement regulations are controlled by the government against violations of labeling and misuse have been difficult to date. Okay. So, um, no genetic modification, but then again, who's going to verify this, really? Um, to the meat supply, clone meat, right? And generally speaking, what sample of organic meat are we going to find somewhere in the wild that we can compare to what they would go ahead and establish as being the assessed sample of organic meat in comparison to the clone meat, which may or may not be genetically modified by FDA approved standards. Procedure risk assessment of meat and milk derived from cloned animals. There are 5 levels in procedure risk assessment of meat and milk derived from cloned animals for guideline establishment approval, survey of human consumption experience or history for long-term analysis, similarities, composition, adverse reproductive effects, genotoxicity. Once again, um, reiterated the same thing, but I'm not really seeing the actual, procedural science of in vitro and also, dish studies and all that. Conclusion. In this paper, I extensively surveyed the risk assessment. It's like, okay, quick. So I extensively surveyed the risk assessment study of animal products derived from cloned animals. The assays for establishment of guidelines are cited for various studies. This author found that there is no evidence that meat and milk derived from cloned animals or their progeny pose a food safety risk in terms of genotoxicity, adverse reproductive effects, or allergic reactions. There are no countries in the world that established guidelines on meat and milk derived, and there's not even a ending dot on the conclusionary statement on was supposed to be something vague. What is it, the preview? Do the preview? Yeah, like I'm not exactly the best with these things. So Oh, purchase FDA. Yeah, like I said, it's one of those. Um, I'd have to register with these. Go ahead and get access to these journals, which fortunately I won't be able to go ahead and get access to them now. But um, yeah, yeah, you always come across these journals where it's like got the meat and potatoes and then some when it comes to the information you really want to know. So let's see. Uh, Extensive evaluation of the available data has. Well, I mean, we got some information, but we didn't really get the information. Generally, just kept on reassessing the overview of what was being posed, but didn't actually give you, generally speaking, any kind of either scientific, medical, or uh, you know, judicial, executive effect of these policies. So we don't have the ethics nor the science or the medical literature, which is behind a paywall. So um, let's see, let's see how much these paywalls are, because let's see, in vitro and vivo genotoxic effects of somatic nuclear transfer clone. Yeah, I mean this is yeah. Um, NCBI is good, but that was the information you're looking for right there. That was it right there. That was it. Uh, just keep looking, Ryan. Keep looking. All right, so um, the antioxidant components of milk towards progressive regulatory approaches for agricultural dust genome editors can be applied to improve meat and milk composition. Whoa. Okay. All right. Towards progressive regulatory approaches for agricultural application of animal biotechnology, right? All right. So, um, progressive regulatory approaches, right? Uh, of animal biotechnology. Okay. Uh, Instead of just skipping right to the conclusion, let's actually take a look here. Tradition, breeding techniques applied incrementally over thousands of years have yielded huge benefits in the characteristics of agricultural animals. This is the result of significant measurable changes to the genomes of those animal species and breeds. Um, Genome editing techniques may now be applied to achieve targeted DNA sequencing alterations with potential to affect traits of interest to the production of agricultural animals in just one generation. New opportunities arise to improve characteristics difficult to achieve or not amenable to traditional breeding, including disease. resistance and traits that can improve animal welfare, reduce environmental impact, or mitigate impacts of climate change. Countries and supranational institutions are in process of defining regulatory approaches for genome-edited animals and can benefit from sharing approaches and experiences to institute progressive policies in which regulatory oversight scale to particular level of risk involved. Countries and conglomerates, international conglomerates, institutions are in the process of defining uh, regulatory approaches for genome edited animals and can benefit from sharing approaches and experiences to institute progressive policies in which regulatory oversight is scaled to the particular level of risk involved. Progressive policies in which regulatory oversight is scaled to okay. So to facilitate information sharing, the discussion on animal biotechnology, an international community of researchers, developers, breeders, regulators, and communicators recently held a series of 7 virtual workshop sessions on applications of biotechnology. Recently held a series of 7 virtual workshop sessions on application of biotechnology for animal agriculture, food, and environmental safety assessment, regulatory approaches, and market and consumer acceptance. Okay. In this report, we summarize the topics presented in the workshop sessions. In this report, we summarize the topics presented in the workshop sessions as well as discussions coming out of the breakout session. This framed within the context of past and recent scientific and regulatory developments. This is a pivotal moment for determination of regulatory approaches, establishment of trust across the innovation through chain innovation across the innovation through chain, from researchers, developers, regulators, breeders, farmers through to consumers. Keywords: genome editing, genetic modification, genetic engineering, public policy, regulatory cooperation, international trade, public acceptance, life. Uh, a number of global trends contribute to the need. A number of global trends contribute to the need for heightened investment in development and distribution of genetically improved agricultural animals. This need is heightened by increased demand for animal-based protein due to growing prosperity and decreasing arable land for agriculture, further ongoing climate. Alright. So genetic modification using rDNA techniques. Historically, the term genetically modified has referred to organisms. So here we're actually getting the actual guidelines. I think this is actually the guidelines here. Let's look at the year. So okay, of course, right, 22, right? I mean, just like Right, right. They couldn't have done this 20 years ago, right? They had to do it just 2 years ago. So, or at least this document is when um those workshop sessions between that whole I guess a think tank or that whole workshop with the think tank of dudes and doodads pretty much thinking about how cloning was a good idea. So I guess see here. Historically, when the those workshops occurred would be good to know. All right, so as well as combining gene ed technique with genomic selection to achieve genetic advances much more quickly than could be achieved by conventional breeding methods. All right, so I'll have to look at this to go ahead and learn what it means. So give me a second. Genetic modification using a rDNA technique. Uh, okay. Hmm. Many promising GM animals have not achieved regulatory approval. For example, the Enviro pig was a transgenic pig synthesizing phytase in the salivary glands and secreting active enzyme in the saliva, enabling them to utilize practically all the phosphorus in cereal grains and soybean meal, and to excrete fecal material containing 60% less phosphorus than non-transgenic pigs. Um, it received environmental approval in Canada for commercial production, but the application for food approval in Canada and the United States was discontinued in 2012. The developer cited the expense, length, and uncertainty of the approval process as key factors for ending the program. Okay. all right. So these are genetically modified um animals ban on import fertilized eggs was lifted in 2019, which is not for sale and consumption in the country. China authority have authorized aqua bounty, regulatory aqua bounty salmon. Chinese authorities have authorized aqua bounty regulatory trials. All right, so the FDA said, you know, they don't genetically modify any of the clone meat they have, right? Um, Generally speaking, I still haven't, um, I'm barely looking into this, so I haven't really looked, I haven't really found any of the information as far as, I think what I was looking for as far as the, was, what it was saying, uh, safety considerations for Americans and so on and so forth and all that. So this is talking about genetically modified animals that are on on the market, right? Now, how do you genetically modify an animal? Well, I'm assuming they've genetically modified the animals in all kinds of ways. I mean, here, so you go ahead and state some of the ways in which they were genetically modified. I'm guessing you can go ahead and take a look at each of any of these particular um, you know, scientific biotechnology developments and uh, and you could see the sort of different methodologies because I mean I'm sure they weren't all the same, right? They're all the same. Here you have CRISPR and all right. So the U.S. Food and Drug Administration recently approved. I'm trying to take a break so I can go ahead and just like refocus my mind here because all right. The U.S. Food and Drug Administration recently approved an rDNA construct inserted into a line of domesticated pigs using homologous recombination referred to as gal-safe pigs. These GM pigs were, I guess GM or genetically modified pigs were developed for biomedical uses and are approved for use both as food and for use in human therapeutics. Other GM animals still in the R&D pipeline include pigs expressing bovine, a lactoglobulin in milk to improve pig growth and health. So mixing cow and pig, and goats expressing lysozyme in milk, which may have human health benefits as well as increased shelf life. While classical gene transfer methods were effective for development of these animals, more so with the advent of somatic cell nuclear transfer or cloning, classical gene transfer methods were effective for the development of these animals, more so with the advent of somatic cell nuclear transfer or cloning. Integration rates were relatively low. Only 1 to several percent of micro-injected embryos subsequently expressed the inserted gene. The integration of the construct was at random sites across the genome, raising concern for insertional mutagenesis. Random sites across integration of the construct. Right. Inserted gene, the integration of the construct was at random sites across the genome, raising concern for insertional mutagenesis and animal welfare. I mean, pretty much pin the tail on the genome. So uh, advent of genome editing. Techniques have recently been applied to agricultural animals to alter DNA. Techniques have recently been applied to agricultural animals to alter a DNA sequence. Of course, it's not, uh, pin the tail on the genome. It's just, the way it's describing it. It's just techniques have recently been applied to agricultural animals to alter a DNA sequence at a targeted site in the host genome using a genome editor to delete. Techniques have recently been applied to agricultural animals. Techniques have recently been applied to agricultural animals to alter a DNA sequence at a targeted site in a host genome using a genome editor to delete, insert, or change nucleotides in the DNA sequence. Genome editors are a set of molecular tools that allows researchers to modify the genome with a higher rate of success and a higher precision than previous rDNA technologies. So there you go. Here's a word for the day. Um, they got mRNA and genetic vaccination stuff like that, right? But here's another word for people to go ahead and actually look at genome editors. Since we got well, there you go. Um, well, different genome editors are available, such as zinc fingered nucleus. Yep. Here you go. Transcription activator like effector nucleases, talons, the newer clustered regularly interspace short palindromic repeats. associated nucleus system, CRISPR, has become the choice of most developers. Um, yeah, so I think we're going to end this study on the biotechnology being introduced into our food supply as far as cloning and um, genetic modification, uh, mRNA vaccination, um, FDA's approval of cloned meat products, but more so states that they actually are, have um, put it on the market for some time now. And specifically, they are saying that the clone meat that they have out on the market is not genetically modified by any means. It's perfectly cloned meat or something like that. You know what I mean? It's cloned meat. That's, um, as they say, it's quite referential to the original material. So at the same time, we have all this genetically modified meat for animals in the food supply. Conclusive statement, none whatsoever. This is a beginning, I guess, um, you know, tutorial discussion on the ethics and established uh, narrative, or the established discussion of pretty much um, what's in the food supply, what's hitting the markets. I mean, I'm not going to speculate because this is way too much to go ahead and start, just speculating this and that just because I want to go ahead and just throw out one cavalieristic statement or the other. I mean, I think there's much more to be said before anything's said at all about what's going on here. But I mean, generally speaking, the narrative is all over the place and it's completely messy. There's absolutely no seemingly apparent ethics or guidelines as far as what has been established to be um, ethics or guidelines. I mean, it just pretty much, FDA approved these things and are now just floating around in the food supply. Now, those journals um, may be much more non-biased in their actual information which they they disseminate. But I'm gonna look for disseminate really quick because sometimes I get these words wrong. All right, yeah, spread again. All right, all right. So, um, I want to get to a conclusive statement about all this today, before I go ahead and just end the discussion. But the thing is that I'm not going to get to it, and I don't want to go ahead and start, stating this or that. It's just, but I would like to, but the whole thing is that I probably shouldn't. Uh, um, really, what we can say, what can be said is that the FDA approved clone meat a long time ago, and we've all eaten clone meat by now, and probably had our fair share of milk from cloned cows. Um, Generally speaking, they say that these are not genetically modified. Now, at the same time, um, there's genetically modified animals floating around in the food supply, which could have bred with the clone meat animals. Um, this doesn't include vaccination, hormonal, uh, you know, hormonal therapy, the hormones they give to the animals and all that. Um, and the mRNA, um, the mRNA vaccination of livestock and animals for the use for the use of food has barely been put in place or sanctioned or ratified. I don't know what's the word. Um, Beginning of this year. So if we ate mRNA treated mRNA uh, vaccinated uh, meat or cheese or whatever, then it's safe to say that um, you know they would label it. So mRNA technology, if it has any mRNA vaccine materials, right? It doesn't state mRNA vaccine technology, and in the meat, then they will put it on the label. if it has mRNA vaccine materials in the meat, but otherwise they won't put it on there. So they won't put labels on the meat if it's from cloned meat. They won't put labels on the meat if it's from genetically modified meat. Um, and they're barely putting labels starting this year as far as mRNA vaccine treated animals that meat is on the market. If it's on the market, starting at the beginning of this year. Like I said, I'm gonna, it's gonna be a while before I can go. I can actually um, get back to a second video on this conclusion. Really, I mean, I I'd rather read the actual information, but okay. Many challenges remain for animal biotechnology to gain trust and acceptance to become available for farmers to use to meet global demand for food products while improving animal welfare and reducing environmental impacts. You notice how they countermeasure both these two, juxtaposed towards each other, even though they throw both factors out the window. Right? Throw the baby out with the bathwater. Thoughtful communication can help consumers become aware. Yeah, thoughtful communication can help consumers become aware of the benefits to themselves, e.g., food prices, availability, enhanced nutrition, uh, how do you call it? Supposed enhanced nutrition, right? I mean, I mean the benefits to themselves, right? Food prices, availability, enhanced nutrition. I mean, these are all terminologies for what they would like to go ahead and state are benefits. Of course, are not actually implicating us like is it going to actually do you harm or be good for you? It's like enhanced nutrition. It's like by all means, be very clear on these specifications because this is supposed to be a medical or scientific uh, journal literature. Uh, and it's supposed to be bending uh, words towards political terminology, which you could see they do it, which I'm not going to get into right now. That's what I was trying to. I was That's what I was trying not to do this entire time. I was like, it's like well, I was gonna read the medical journals, not start criticizing and start establishing some of the political lingo they put into the yeah. I mean, how they make it arbitrary. So, the environment improved efficiency, reducing inputs in greenhouse emissions, and so forth. Give adoption. Yeah, dude. Yeah. This is it's kind of like the end for this session as far as this because mean, I read the conclusion, and I want to actually I want to read the actual journal before I start actually making any actual statements about what is going on here. But we'll read the conclusion just to go ahead and end the video. Many challenges remain for animal biotechnology to gain trust and acceptance and to become available for farmers to use to meet global demand for food products while improving animal welfare, reducing environmental impacts. Thoughtful communication can help consumers become aware of the benefits to themselves. Food prices, availability, enhanced nutrition, animal welfare, reduction of diseases, cattle that will not need to be dehorned, pigs that will not need to be castrated, and the environment. Improved the efficiency, reducing inputs in greenhouse gas emissions, uh, aquaculture production, reducing demand on natural fisheries and using fewer resources, as well as cost of inaction, prevalence of preventable diseases, environmental cost, degree of adoption of animal biotechnology will be determined by public perception and the development of regulatory approaches that are science-based and risk proportionate. Many critics of animal agriculture and biotechnology emphasize the need to reduce or cease the use of animals due to environmental impacts and welfare concerns. Notice how they keep on averting the entire narrative towards the environment rather the uh, predisposed possible risk and health hazards towards the individual, and denoting specifically the scientific notation as far as any kind of conclusion as far as this scientific and scientific discussion. These concerns are valid to some extent, however, demand for animal products will continue and will increase in some regions for the foreseeable future. Genome editing technology offers an opportunity to rapidly improve animal welfare and reduce environmental impact that would otherwise be prohibiting using conventional breeding, and if not cost prohibitive, could take decades to achieve. I'm not going to even make any statements about how what the other um, uh, medical literature was stating and implications to this one, because I really want to read this before I go ahead and actually give some actual commentary. The community of professionals that participate, but to be clear, um, the other one, stated clearly that it would mostly be, um, biotechnology companies and for the sake of academia and so on and so forth, universities, institutes, research facilities, so on and so forth, uh, who are going to take advantage of this, who are going to use it, and it's not going to be for the uh, imposed statement of it being for these purposes as stated here. given these statements are 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 given are appropriated. They're given any appropriation. If they're not given any appropriation, then of course, we're looking at a major welfare concerning all of us, pretty much being um, not established. So many challenges remain for animal biotechnology to gain trust and acceptance and to become available for farmers to use to meet global demand for food products while improving animal welfare, reducing environmental impacts. Thoughtful communication can help consumers become aware of the benefits to themselves, food prices, availability, enhanced nutrition, animal welfare, reduction of disease, cattle that will not need to be dehorned, pigs that will not need to be castrated, and the environment improved efficiency, reducing inputs and greenhouse gas emissions, aquaculture production, reducing demands on natural fisheries and using fewer resources, as well as the cost of inaction, prevalence of preventable diseases, environmental cost, the degree of adoption of animal biotechnology will be determined by public perception and the development of regulatory approaches that are science-based and risk proportionate. Notice that it says determined by public perception and not public. Um, just public perception instead of public agreement. I think public agreement, right? Public discernment, like by public. I want to get through this thing, man, but like it gets to these certain things right here, where it's like will be determined by public perception rather than public's consent of transparent. All right, I mean, like it's public's perception and development of regulatory approaches that are science-based and risk proportionate. Many critics of animal agriculture and biotechnology emphasize the need to reduce or cease the use. Public perception, right? Determined by public perception. Degree of adoption of animal biotechnology will be determined by public's perception of the transparency of the science uh, the and development of the science, right? Transparency of the science. Um, to establish the regulatory approaches that are science-based and risk proportionate. But anyways, it says this, determined by public perception and development of regulatory approaches that are science-based and risk proportionate. Many critics of animal agriculture and biotechnology emphasize the need to reduce or cease the use of animals due to environmental impacts and welfare concerns. These concerns are valid due to some extent, however, demand for animal products will continue and will increase in some regions for the foreseeable future. Genome editing technology offers opportunity to rapidly improve animal welfare and reduce environmental impacts that would otherwise be cost prohibitive using conventional breeding and if not cost prohibitive could take decades to achieve. The community of professionals that participate in the virtual workshops identified steps that might be taken to achieve the conditions needed for GM and GN ed animals to be used in practical application in agriculture. Building human infrastructure is critical for effective regulatory oversight. Regions developing regulations need to train scientists on regulatory systems and communication, not only to support oversight of animal biotechnology and to create the next generation of regulators. Training is needed for assessing animal biotechnology, animal welfare, and food and environmental safety. Facilitating international dialogue and training is especially important in regions that are developing regulations. Continuing communication between scientists, developers, breeders, farmers, regulators, and public, and the public, right? Continue communication between scientists, developers, breeders, farmers, regulators, and the public is necessary for all sides to build trust and transparency. Assisting journalists, teachers, and politicians in their understanding of the basics of biotechnology and value of its application is essential. Workshops for training scientists in public communication, as well as regional and global workshops to disseminate information on research, regulations, and communication best practices are ongoing or under development. Communication strategies must be designed for particular countries and regions based on the needs and concerns of the regional public. Regulatory alignments or compatibility and modernization of existing regulations are important to enable development and trade um, while encouraging innovation. Regulatory alignment, at least within and amongst some regions, would reduce non-tariff barriers to trade that limit growth of the industry, particularly for smaller companies. Regional workshops and meetings will allow continuing information sharing, especially of regulatory guidelines and case studies of successful and unsuccessful application, especially regulatory guidelines and case studies, successful and unsuccessful application. Acknowledgements. We gratefully acknowledge the contributions of presenters and participants in the virtual workshop series on regulatory approaches for agricultural applications of animal biotechnology. There it is. These workshops were the products of collaboration among the U.S. Department of Agriculture, ISAAA, ISA, and the International Service for the Acquisition of Agrobiotech Applications, IICA. The the International American Institute of Cooperation on Agriculture in Virginia, ISA, the International American Institute for Cooperation on Agriculture, and Virginia Tech University, and supported by agreements with the USDA Foreign Agricultural Service. JPB was supported by an AAAS Science and Technology Policy Fellowship. Served at the USDA Office of the Chief Scientist. The manuscript was strengthened by the attention to comments of two anonymous peers and the editor-in-chief. The manuscript was strengthened by the attention to the comments of two anonymous peers and the editor-in-chief. Glossary of terms. funding. International workshop was supported by the U.S. Department of Agriculture, Foreign Agricultural Service. Availability of data and materials. Supporting material may be found at here. Okay. Author Mark Walton is employed at a company commercializing a GM animal. All author of the other are by academic research, government, non-government organizations, or international cooperation agencies mentioned by any company or product is for informational purposes and does not represent endorsement by the U.S. Department of Agriculture. Mention of any company or product is for informational purposes and does not represent endorsement by the U.S. Department of Agriculture. The findings and conclusions in this publication are those of the authors and should not be construed to represent any official USDA or U.S. government determination or policy. The findings and conclusions in this publication are those of the authors and should not be construed to represent any official USDA or U.S. government determination policy. So this is a study, a funded study, medical scientific journal, biotechnology of genetic engineering animals, and the workshop that it was referring to is this one right here. Uh, the virtual workshop series, right, on regulatory approaches for agricultural applications of animal biotechnology. So there you go. That's the workshop we're talking about. Regulatory approaches for agricultural applications of animal biotechnology. This is what you want to look at. Technical methods applied in animal biotechnology have evolved, and GNED techniques are becoming widely applied to develop animals with new traits. Whatever the biotechnology approach, there is a widely perceived need to assure the public of the food safety of products from genetically modified animals. National and supranational bodies have worked to develop policies for that purpose. Many have done so for organisms developed using classical rDNA technology methods. Additionally, most countries are signatories of the Convention of Biological Diversity um, and the Cartagena Protocol on Biosafety. The regulatory framework generated to govern transboundary movement of living modified organisms and to ensure the human health and environment safety. And development of GNED techniques is relatively recent, and therefore many countries' supranational bodies have yet to determine their regulatory posture. It's noted. Yeah, yeah. So this is the reason for those workshops, which are noted at the bottom of this document, which is there in the acknowledgments. Um, Go ahead, take a look, because I'll have to read through this thing, man. So it's not gonna be just, I'm come back to you in about an hour. It's like, all right. 2020 online workshop. Summary 2020 workshops. Animal biotech resources. Animal biotech resources. Animal biotech resources. Uh, Seems to, okay. Um, hmm, okay. All right. All right. So you would also need access to the actual workshops to see what was actually being discussed. There seems to be plenty of information here. Seems to be alluding towards discussions that were going on, but I'm not going to look into this right now. Yeah. Um, that concludes our study on um, study on uh, genetic, I guess, animal cloning in the food supply, genetically modified food animals in the food supply, uh, mRNA treated, vaccine treated meat and animal products in the food supply, and uh, and that concludes our study for today.